Welcome everyone to Alex Duman's dissertation defense. I'm his PI, Manny Azizi. It's been really wonderful having Alex in the lab the last 5 years. As most of you know Alex, he's a real go-getter. He gets things done. He came here with really good skills already, but he's always been committed to acquiring more skills and learning along the way. In that way, he's a real scholar. And he understood that the PhD is more than just the specific topic in which you gain your expertise, but that it's an opportunity for you to really teach yourself how to acquire knowledge efficiently and effectively. And he's done that really nicely. His scholarship is great, and I've really respected his ability to not get down when results don't go your way. As you'll see, a couple of those chapters didn't quite come through the way we thought they would. And he's absolutely unafraid of taking on hard things. And so he trusts himself to be able to learn it, figure it out, and solve the problem. And he's done it independently. I've never felt less useful for any students than Alex. He just would get things done before I even thought of them or asked him to do it. In addition to his own scholarship, I think he's been a really great member of the community, helping out lots of other students whenever he gets the chance, whether that's sharing code or sharing knowledge. He's just been a really important member of our group. And then finally, I think Alex really has set a new bar for what a TA can do in a class. In my opinion, he's been the best TA I've ever had. And he combines 3 important things. He understands the course structure, mechanics, and the tech that goes into running a course. He understands the material inside and out, and he's excellent at communicating with students. So to me, he's really set a new bar in terms of being an excellent TA. It's going to be sad to see him go, a big loss, obviously, but I know that he's going to succeed in whatever he does just because he's bright, hardworking guy, as you'll see in his talk. So with that, I'm going to turn it over to Alex Duman.
All right, thank you for that introduction, Manny. Thank you, everybody who's here in person and on Zoom. So today I'm going to talk a little bit about my dissertation, which is really moving in a complex world, how surface mechanics and proprioception actually influence our locomotion, the way we move. And before I get started, it's not really playing right now, unfortunately. There we go. I want to give a shout out to Patrick Liu, one of my friends in the department who actually helped me get a lot of these shots that you're seeing here. I also want to give credit to my 2 lab mates, if this will connect and move forward. So they created this really great caricature of me that you can see that ended up being on the advertisement or flyer for today. So I want to thank both Elizabeth Mendoza and Adrian Arias for making this. So for those of you who don't know my background that well, I'm a really avid runner. I used to run in cross country and track in middle school and high school. So in cross country, right, we're mainly running across grass or trails. So really kind of compliant or in some cases soggy conditions. I also did road races where you're running across road, right, or pavement. So really rigid surfaces. And then in track, right, you're running across this surface that's pretty rigid, but has some give to it. So I've already experienced all these different surface conditions, right? And this isn't a problem that just humans face when moving. This is a problem that all animals face in their natural environment. So you can see some animals are locomoting across really compliant surfaces, right? Like soft mud, or even moving across the surface of water in some cases, right? So it's gonna have a lot of give to it. And other animals move across really compact or rigid surfaces like compact earth, rock, or right cement, like in urban environments. So I'm really interested in how this surface stiffness or compliance actually affects the way that animals and we move. So in order to study that, I actually used a model organism, the cane toad, which you can see here. And just for reference, this cane toad is about the size of your fist. So they're a pretty small toad. And they're actually native to Central and South America. So that blue kind of purplish region there, but they've been introduced throughout these different regions in the world in red to control for the cane beetle, where they get their name from. And because they're amphibians, they traverse a wide range of conditions or environments, right? So they can be moving through these really soggy, muddy swamps where there's going to be a lot of compliance in those surfaces that they're landing on, as well as moving through fields or potentially forest floors where there's going to be leaf litter that might dampen their movement. And then, of course, they're moving across rocky creek beds, or in this case, right, they're traveling across roads in Australia. So they're dealing with really rigid surfaces as well. So toads don't walk or run like we do. They actually jump or hop around, like you can see in this video in the center here that's playing out. And they're actually jumping multiple times their own body length. So if we were able to jump like a toad, we'd be able to dunk a basketball from the 3-point line as easy as if it was a set to us. So you can see I'm plotting the actual impact force or ground reaction force on the right. So I'm going to pause that jump there and we can look at this impact force. So this is just the amount of force that toad is experiencing upon landing. And we can see that it actually generates over 3 times its own body weight and force over the course of that landing. So they really have to deal with these large impact forces at touchdown. In addition to that, the most research that has been done on these animals and in most animals and humans has been looking at the takeoff. So that acceleration phase, or when you're putting energy into your body, right, to take off. And there hasn't been a lot of research on that landing or that deceleration phase. So I'm really interested in studying that landing behavior. So as we saw in that video that we played before, their arms are really tuned for that landing behavior and their legs are really tuned for that takeoff. So unlike bipedal organisms like you and me, where we jump with our legs and also have to land and dissipate that energy with the same legs, these animals can essentially evolve different muscle properties in their legs and their arms for those 2 different tasks. And so we actually see evidence for this in pre-existing literature. So this is a figure from Cox and Gillies 2015 that's showing you the jump distance along the x-axis. And it's showing you the elbow angle at impact, so how essentially extended their limbs are. And so we can see as that jump distance, that x-axis increases, the elbow angle is actually also increasing at touchdown, and that makes sense, right? They're trying to get their arms out further on these longer jumps to dissipate a greater amount of energy from that longer hop. So I'm, again, really interested in how surface compliance might affect these animals. So no one's really looked at that before. So we can at least think about how we might land on different surface conditions, right? So you might choose to land differently on a rigid surface that's like concrete, right? It's not going to move at all, versus something that's springy or it has some compliance to it, right? And so there's actually been some research on this as well before in humans. So Ferris and Farley back in the 90s did some work that basically modeled how we land on these different surfaces by treating our leg as a spring and condensing all the mass of the body down to that little blue point right there. And they found that when you land on a rigid surface, you actually have more compliance in your leg. You actually compress more, versus when you're landing on something that has some give or has some compliance to it already, like that springy surface on the right there, you actually keep your legs stiffer, right? And you can push that surface down and allow that energy to be dissipated by the surface. So I'm interested in how cane toads are affected by this surface property. So this brings me to my first chapter, which is how that surface compliance really affects their coordinated landing behavior. So the driving question here is, does that substrate stiffness actually affect their coordinated landing behavior? And we're interested in 3 kind of things. So the first thing is the kinematics, how they're actually moving their arms during this landing period. And if that's going to change whether they're landing on something that's rigid or something that's springy, that's shown here. We're also interested in the kinetics, so the forces, that impact force that they're going to have, are they hitting with the same amount of force on a rigid surface versus that springy compliant surface? And then lastly, right, we were talking about energy, so we want to know how much work or energy the arms are doing in order to effectively land and decelerate the body on these rigid and compliant surfaces. So in order to test that, I created this platform that's actually split in half. So you can see on that right half, I have a force plate in there, and that's actually measuring the impact of the ground reaction force vector, which is shown there with that blue arrow from the toad. And so this whole platform can actually freely move or slide vertically up and down because it's attached to this linear translation stage on the far left. And so we can actually lock that platform in place with a bolt. So when that toad comes in, it can apply however much force it wants, but it's not going to move. It's going to act totally rigid. We can also unlock or unbolt this platform, and now it can move, and we can actually attach springs to it so that when this toad comes in and applies some force, it's going to stretch that spring and actually displace that platform a given amount based on how stiff that spring actually is. So that's essentially what I did. I created 4 compliance treatments that I was interested in testing. I randomized them in order, which basically means the toads have no idea if they're going to land on something that's rigid or something that's really springy and compliant. So those 4 conditions that I have, it's 0, 2.5, 5, and 10 millimeters of displacement per body weight. So really this 0 millimeters of displacement is going to be this toad coming in and landing on something that doesn't move at all. So it's basically acting like concrete. That surface doesn't move. And on the other end, that 10 millimeters of displacement, that's a full centimeter of displacement for every unit of body weight of force that they're applying on this surface. And so you can think of this as a really compliant surface, like maybe a trampoline. And because these toads, right, are the size of your fist, their arms are actually only about 5 centimeters long. So that 1 centimeter of displacement per unit of body weight is actually pretty significant. This would be like if we jumped onto a trampoline and you sunk almost halfway up to your knee, right? So really compliant surfaces. So I had 8 toads that I basically landed on all 4 of those conditions, and I got 5 replicate landings on each 4 of those compliance treatments. So like this example you can see here. So let's take a look at just an example. So 2 trials of what the same individual is doing on the rigid landing on the left and the most compliant condition on the right. So you can kind of see that landing behavior there in the videos. I'm actually going to pause this so we can look at the graphs. So this top graph, or that top panel in orange, is the limb extension ratio. So essentially what that is it's going to approach 1 as the limbs become fully extended, and it's approaching 0 as they become more flexed or compressed. And so we can see early on in the landing phase, those limbs are really extended out, right? Because when they first touch down, they're trying to keep their arms extended so they can dissipate this energy and slow down. And then over the course of landing, right, they're applying some force and some work in order to slow themselves down. So we see that the limbs compress, and they actually compress about the same. Those traces look similar between these 2 different compliance treatments. And then down in the center panel in blue, we actually see the magnitude of the impact force, or that ground reaction force in blue. And again, the rate of that force buildup and the peak are actually the same between these 2 trials, right? So they're kind of coming in with the same force on these 2 landing conditions. And then in that same plot in the center in gold or kind of yellow, we actually see the platform displacement. So this first trial on the left, right, we set as a rigid condition, so we don't expect it to move at all. And that's exactly what we see. This yellow line is basically staying at 0 the whole time. And on the right, this is a really compliant condition. So we see that platform actually is accelerating, and eventually it reaches a displacement of 6 millimeters by the time that the legs come down in contact. And then in the bottom graph, we actually have the energy or the amount of work that the arms are doing to slow the body down. And this is cumulative, so it's building up over time. And again, we see it's pretty similar across these 2 different compliance treatments. So something that I realized after I collected all this data was that this platform is really large. So the mass of the platform, you can kind of see down there in red at the bottom, is 725 grams. The average mass of our toad is only around 120 grams. So it's almost 10 times larger, this platform, than the mass of the toad. So you can think of this really small, like toad, trying to move this really big object. It's actually similar in comparison to a fully grown human like myself, trying to push a small car. So what I did was I essentially tried to create a second platform that was really small, and I actually stripped down as much weight as I could, and I got it to around the average mass of the toad, 123 grams. So instead of pushing a car, this would be like you're pushing your friend down the street in an office chair, right? So you can still get the same amount of displacement, like I can still push my friend in the car the same distance, but it's going to take a lot longer to push that more massive car because of the inertia. So again, we're going to look at just 2 representative trials. So here's the same individual landing on a smaller platform on the left and the more massive platform on the right. These are actually both the same compliance treatments, so it should displace about 5 millimeters per unit body weight of force. And I'm going to pause it again just so that we can kind of look at these graphs. So again, the limb extension ratio really looks the same. You're starting off really extended and compressing similarly across these 2 different sizes of platform. The ground reaction force, that blue center graph, right, still maxes out at about the same value between these two conditions. And at least initially, that rate of force increase is the same between the two trials. But really what we're keying in on here is that yellow or gold trace, right? So that line represents the movement of the platform. And in the smaller platform, we actually see that it displaces to that 5 millimeters about halfway through a landing period at about 60 milliseconds. Whereas in the larger platform on the right, it takes almost that entire landing period to reach just that 5 millimeters of displacement. So again, it's taking much longer to move that larger platform the same distance. And then at the bottom, again, we're seeing the cumulative amount of work or energy that the arms are dissipating. And that actually looks pretty similar across these two platform sizes. So let's not look at individual jumps anymore. Let's look at all the jumps kind of together. So this is actually a rain cloud plot. I know it looks kind of busy, but I'll walk you through it to kind of understand how to interpret these. So on the x-axis, we have the compliance treatments that we chose. And on the y-axis here, we're actually looking at the maximum platform displacement we observed during that landing. So we'll just focus in on the 2.5 compliance treatment for now. So we kind of see these two classic bell-shaped curves that you notice. One's orange and one's kind of this yellow color. So those two different colors represented two different sizes of platforms. So that darker kind of orange is going to be the larger platform, and that lighter yellow is going to be the lighter or smaller platform. And so these kind of bell-shaped curves, right, represent that distribution, or they're the cloud of this rain cloud plot. And at kind of the base or the right side of the clouds, you have this big central dot, and that represents the mean for that platform size. And then there's these little whiskers or error bars that represent the 95% confidence interval of that mean. So we're really confident that that mean is found somewhere in that region. And then just to the right or underneath those clouds, we have the raindrops. And so those raindrops actually represent the individual landing trials that we observed for all the individuals. And here what we can see is that these distributions do tend to increase across the x-axis, or as we increase the compliance, or how much give that surface has. And so that's exactly what we expect, right? Because we want the platform to displace a greater amount as we increase the compliance of those surfaces. So this graph is essentially showing us, right, that those springs, the conditions we chose, are actually producing different displacements, which is what we wanted, right? But we don't really care about what the platform is doing. We care about how the animals are moving, right? So here what we're looking at is that change in the limb extension ratio over landing. So you can think of this as the arm compression during touchdown. And these distributions overlap quite a bit, going left to right across those compliance treatments. And again, their averages kind of all line up as well horizontally. So here, there's really no significant change in how they're compressing their limbs across compliance treatments. So really, these toads are landing the same way on the rigid surface on the left as they are on the more compliant surfaces on the right. We also looked at the kinetics, right? So in this case, it's the maximal impact force these toads are experiencing. And again, we see the same trend. They're not really changing the peak amount of force that they're impacting with across these compliance treatments. So really, they're impacting with the same maximum force on that rigid surface like concrete as they are on the springy trampoline. And then lastly, we looked at the energy, right, that the arms are dissipating and the amount of work that they did to slow the body down. And again, it doesn't seem to differ across compliance, although it does seem like they're doing a little bit less work on that smaller platform. So clearly, it's not taking any amount or more muscle, I should say, to land on the different compliance treatments, but they're using more work or muscular effort, right, to land on that larger platform. So you can kind of see they have a little bit more muscle to them there. So the main takeaway from this first chapter is essentially that toads are landing the same way on that rigid surface as they are on the compliant surfaces. So probably not how you and I would normally land on like concrete, right, versus a trampoline. So this kind of was a weird result, and it got me thinking, are they actually using their muscles similar, right? Because we randomized the order of these surface conditions that they were going to land on. So are they tuning their muscles in the air the same, or are they reacting after they've landed and potentially had time to experience whether they're landing on something like concrete or a trampoline? So this brings me to my second chapter, which is how the cane toads are really controlling their muscles in order to coordinate an effective landing. And here the question that I'm asking is, are the toads, right, using those muscles the same across these different surface conditions? So you can see this video, this is the same kind of setup I used in chapter 1. So I'm still using that same platform from chapter 1. But now we're really interested in these muscles and how they're being activated. So I'm going to measure the EMG, or the muscle activity, in these 4 muscles. So we have the deltoideus, which helps bring your shoulder out and forward. You have the pectoralis, which we're all probably familiar with doing push-ups, right? It kind of brings your arm in and forward as well. And then the anconeus is essentially their triceps muscles, so that helps with elbow extension. And then lastly, the palmaris longus is a wrist flexor, so it kind of brings the wrist out and helps brace for the impact. So I had 5 animals that I measured these EMG, or muscle activities, from. And here's, again, just 2 representative trials of the animal taking off and landing on the rigid surface on the right, and landing on a compliant surface on the right. Sorry, I might have said left and right wrong. But so I'm going to pause this after it's done playing, and we can take a look. So again, I just have the limb extension ratio through time. And now we're not just looking at the landing phase, we're actually looking throughout the entire jump. So this first kind of white region on the left of all the graphs is going to represent when the arms are still in contact with the ground and the toes really just getting ready to take off. This kind of bluish gray box is actually when the arms are up in the air, and that's potentially when the toes are getting ready to land or prepare for that landing. And then that last little white box at the end, that's when the arms are back in contact with the ground and doing the actual work to slow the body down. So we can see actually in both these trials, all the muscles are really active in these blue aerial phases. So when they're in the air preparing, they're turning on all these muscles to prepare for that landing. And then even after the landing, you see all these muscles are actually quite active, right? Because that's when they're doing the work to slow the body down. So let's just focus in on one of these graphs to kind of understand how we actually analyze this EMG, or muscle activity, data. So we're going to zoom in on the anconeus. And essentially what we do is we take the absolute value of this curve. So that makes sure everything is positive. Then what we can do is actually take the integral, or count up all the area underneath that curve. And that gives us a sense of the intensity, or the amount of activity that that muscle has been on during that period of time. We can also, right, see how long that muscle has been on. So maybe we're really interested in how long this muscle is on in the aerial phase, right? To see how long they're actually turning on their muscles to prepare for that landing. So that we call the active duration of the muscle, right? And we can actually also measure the intensity, so the area underneath that curve, just during the aerial phase, if we're interested in things like that. So that's exactly what we did. So here we're looking at the EMG, or that muscle activity, just for the deltoideus that you can see up top, across compliance. And this is just right for the aerial phase when they're preparing for that landing. And you can see again, these distributions look almost identical across those compliance treatments. So at least in the air, they're not tuning that deltoideus muscle that kind of helps bring their arm out and forward any differently based off the landing conditions that they actually haven't really even experienced yet. So we wanted to check right after landing, do they change it at all? And again, these distributions look almost identical, right? So they're really not even tuning that deltoideus muscle after they've landed either across these different compliance treatments. And so this trend of not seeing any significant changes in how they're activating the muscle was actually what we saw across all 4 of the muscles that we looked at. So then we got to thinking, okay, well, maybe if they're not responding to like the landing that they, right, it's randomized and they might not know what to expect, maybe they're actually landing or tuning their muscles based off of the previous jump that they just landed from, right? And so we analyzed that, and we actually found that the previous trial also had no effect on that muscle activation, this intensity here. So the graphs kind of also looked identical. So here the conclusion was that toads are essentially tuning their muscles similarly, right? Again, when these conditions are essentially unknown to them, they're randomized in order. So they don't necessarily know if it's going to be a rigid or compliant surface. And this also, right, kind of mimics those results that we saw in my first chapter, where they're really moving their arms and hitting with the same amount of force, so across these different compliance treatments. So there's 3 kind of strategies I've thought of that they potentially could be using to kind of perform or behave this way. And if you think of others, like please let me know, because I'm always curious. But the first strategy is that they're essentially always assuming that this surface is going to be rigid, right? And so that's probably the safest, right? Because the rigid surface is going to take the most energy in order to effectively slow yourself down and land. You probably don't want to crash into a really hard surface, right? Because that's when you're going to get injured. And even if you land on something that's compliant, if you're ready to land on something that's rigid, you're probably going to still be able to land on that surface effectively. The second possibility is that they're not just remembering that most previous hop, they're remembering several of their more recent hops. So it could be that we need to hop them several times on this compliant surface kind of continuously before we even see them starting to tune that muscle activity. And then the third thing is they could actually be predicting that landing surface based off the conditions they experienced during their takeoff. So in both my first two chapters, they actually jumped off of a rigid surface. So they're experiencing concrete when they're taking off. And so it could be when they're in the air, right, preparing for that landing. And once they touch down, they're actually thinking back to that takeoff condition and remembering that rigid surface and anticipating that, right, preparing for that rigid surface. So there actually is some evidence for this in the pre-existing literature. So here there's a graph from Cox et al. 2018. That's basically showing you the elbow angle on the y-axis, so how extended this elbow is through time. And here we're going to kind of focus in on that black line with the kind of gray outline areas. So that's normally what a toad does. So I kind of sketched it out above if that helps, but I'll also walk you through it so you can watch it on me. So when toads first start to jump, their limbs, their arms are going to start pretty extended because they're just lifting up. So you can see this angle is pretty high to begin with. And then, right, as they come up into the air, they're going to start flexing and bringing their limbs up. And so that you can see brings that elbow angle down really rapidly in the first half of that graph. And then at some point in the air, they're actually going to start to extend their limbs and increase that elbow angle before they touch down. And that's that second half of that plot, right? You see this really steep increase in that black line. So what this study did was they actually cut the sciatic nerve, so the nerve that runs down the length of the leg or the hind limb, right? And so now all the muscles in the leg can no longer really tell the arms or the rest of the body what they're experiencing, right, during that takeoff. And we can see that data in the dotted kind of red or purplish line there. And we see that those animals actually still extend their arms or their elbow throughout that aerial phase, but they're extending at a much slower rate. That slope is much more shallow than we see in the normal toads. So I should also mention that this study also looked at whether vision or the vestibular sense, so that inner ear, your sense of balance, had an effect on this landing behavior. And it really didn't have as great of an effect as cutting that nerve and blocking the information coming from the legs. So this brings me to my third chapter, which is how that proprioception or sensory information from the legs actually might influence the arm behavior and preparation for their landings. So this proprioception or sensory information, we're probably more familiar with it being called the stretch reflex. And you've probably had this test done on you before if you've ever been to a doctor and they've tapped on your knee, right? You notice that your foot kind of just kicks out without you even knowing about it, right? You see it after the fact. So what's actually going on here is that the doctor's tapping on tendons that are connected to your quadriceps muscle, and that really rapid tap is actually stretching the muscle or lengthening it really rapidly. And your muscles want to avoid being stretched or lengthened, right? Because that's when we pull a muscle, right? Or it becomes damaged. So if we zoom in at the level of the muscle cell or fiber, we can see there's kind of different sizes of muscle fibers. And some of these smaller muscle fibers actually have neurons that kind of wrap all the way around them, and they'll actually sense the length changes in this muscle. So when your doctor taps on that tendon, right, it stretches the muscle, it stretches these cells and actually stimulates those neurons, and they send this information up to the spinal cord, which you can see. And in the spinal cord, that information gets relayed to motor neurons, which then send a command back down to the other muscle fibers or cells in the body or in that muscle and tell it to contract, right? So it's going to shorten and try to resist that lengthening of the muscle so that it doesn't get pulled apart or injured. And so this whole pathway is the stretch reflex, and it can actually get disrupted by things like physical injury or damage to the neurons, or damage from complications from illnesses like diabetes. And generally what happens is, you can see up in the top right, the muscle spindles don't really function properly any longer. So you lose out on that length sensitivity of the muscle, but the motor neurons work just fine. So you're able to move around and walk usually pretty normally after injury or from this diabetic-induced neuropathy. And so this is actually the mechanism we think might explain why their arms, right, are moving differently. Because when they cut the nerve in the leg, right, they lose out on this stretch information from that muscle, that length change, and that could be important for informing the arms on how to behave. So I did a surgical intervention in order to test this, but before I did any surgery, I actually took 11 toads that were totally intact, so their nervous system is still functioning normally, and they served as our control. So you can kind of see I've zoomed in on their thigh right there, and we can see the sciatic nerve, right, runs down the length of the leg, and essentially innervates all the muscles in our leg and tells them how to move. So the surgical condition that we actually care about is this nerve reinnervation procedure that I did. So I created an incision along the length of the thigh and basically cut through all the connective tissue down to the bone where this nerve runs along. And you can cut that nerve or transect it and then suture it back together. And what's really cool is when you put the two nerve ends back together, it'll actually heal. And so those motor neurons that I talked about that come down from the spinal cord and tell the muscles how to move, those heal back just fine. But those muscle spindles that send the information from that muscle back up to the spine never fully recover. And so because we cut through all this connective tissue, I want to make sure that that's not causing any influences. So in 4 individuals, I did a sham surgery where essentially I did the same incision, cut through the connective tissue down to that nerve, found the nerve, but then didn't touch that nerve at all. So their nervous system should still be intact. So in order to measure the actual stretch reflex, I had to come up with a different kind of test than just tapping on their tendon, because toes are pretty small, right, the size of your fist. So I developed this little system that's basically just a hinge connected to the table. And at the top of that hinge, right, is, well, they're actually sitting in this, right? So strapped in their ankle in this case. And at the top of that hinge, there's a string that's connected up to a motor. And this motor can apply a really rapid little tug on their ankle and cause it to flex, right? So it's going to decrease that angle. And so you can see that on the graph at the right in yellow. It's kind of just sitting still for a while. And then in that gray region, the motor's actually turned on and it's causing really rapid ankle flexion. So it's decreasing that angle. And what that's going to do is it's actually going to stretch those calf muscles, right? If you try to pull your toes towards your shin, it stretches that calf muscle. And right when we stretch those muscles, they're going to try to resist that and turn on and actually contract. And so that's what actually is driving this blue phase, the ankle extension, is when the muscle should be really active. And so we measured the EMG, right, that muscle activity in red here from that calf muscle. And you can see it's really not that active when we first start to stretch it, but it really kicks on when that ankle is extending, right? And that muscle is actually turning on and driving that ankle extension. So what we did was we actually quantified the intensity or the area underneath these curves, right? And we did it in the extension phase, so that kind of blue area, right? So it's the area in this blue area relative to during the flexion phase, so the amount of area underneath that red curve in that gray box. So we can see these pre-surgery individuals that have a totally intact nervous system have a ratio about 20 times more activity in that blue ankle extension phase. And this is showing, right, that the nerve is working. So we also measured this after our sham surgery. And again, that ratio is really close to 20. The error bar is going to be really big, right, because we only have 4 individuals, but it actually doesn't significantly differ. And so again, this nerve appears to be working, right, and the nerve system's intact, so that makes sense. And then after I did that nerve reinnervation surgery, where I actually cut the nerve and allowed it to heal back together, we measured that same ratio, and we saw that ratio actually drops to 1 week after the surgery, all the way up to 6 months following that surgery. And keep in mind, a ratio of 1 is actually what we would expect if we see that random noise that kind of starts at the beginning to continue on for both those phases, right? And just in case you think there's actually a signal going on there, I want to kind of show you the difference between a 20-fold increase versus a ratio of around 1. you probably can't even see the text that I've already put up on the screen because it's font size 1 versus font size 20 of this nerve is working, right? So even if that information is getting to the spinal cord, it's probably not going to be able to respond effectively, right? And produce a signal, right? And if that's still not proof enough that we've knocked out this stretch reflex, I also measured it after the toads were euthanized. So the nervous system is no longer working because the animal is no longer alive. And post-mortem, again, that ratio drops to 1. So clearly when the nervous system's not working, this is what the ratio should be. So we really showed that here we knocked out that stretch reflex. So that stretch reflex is gone. It actually stays out. We never really fully recover it, even 6 months after this surgery. So again, we don't really care about this stretch reflex. We just want to show that they've lost this information. What we actually care about is how these toads are moving when they've lost that sensory information. So here, what we actually measured was the hind limb extension, because we want to know how that hind limb is moving to make sure that they're actually jumping the same way after we've done the surgery on their legs. And really what we care about is the elbow extension, right? Because we saw in that previous study that they actually slow down the rate, they extend their elbows. And we're also interested in how the forelimbs are moving as they're hopping. So 6 months after the surgery, I also implanted EMG electrodes so we could measure the muscle activity from their plantaris, so that calf muscle I mentioned earlier, and then the anconius, right? So that's that muscle we saw before that extends the elbow and kind of preps the arm for that impact. So here what we're looking at is the average rate of their leg or their hind limb extension during takeoff. And again, this rain cloud plot shows that these distributions overlap quite a bit in the pre-surgery individuals on that far left, so before any surgery is done, and the 4 sham individuals, so the nervous system is still intact in the center plot, and then those 6 months post-surgery, right, where this nerve has been cut and they've lost out on that sensory information. So really what this shows is those 2 surgery conditions that we did really did not affect their hind limb whatsoever. They're still jumping relatively similarly. And then really what we're interested in, right, is that arm behavior. Are they moving their arms differently? So we looked at the average rate of their elbow extension in preparation for landing. And so we actually see there's a significant drop from the after these pre-surgery individuals in the sham and the post-surgery, so when that nervous system isn't working. So essentially what this is showing is that after these 2 surgeries, the toads seem to be moving their arms or extending their limbs more slowly. And we actually don't think these sham individuals are really truly moving their limbs slowly because of the surgery. I actually did the sham surgeries first, and because these were my first surgeries, right, on really relatively small animals that are the size of your fists, I wanted to make sure I could find those nerves, so I used the largest individuals, right? And we already know that larger toads can actually jump further, and they actually did jump significantly farther than the individuals in the pre and post, which are essentially the same group of toads. And so we really think, right, these toads are essentially larger, jumping further, and therefore in the air for a longer period of time, and can get away with extending their limbs a little bit more slowly. So we think it's a size or the size of the toad that's actually driving this significant decrease in the sham, and it's the loss in that sensory information driving this decrease in the post-surgery individuals. So we also looked, like I said, at the muscle activity. So in this case, we're not looking at the intensity, we're looking at the duration. So the amount of time that that anconius or the elbow extensor muscle is on in the air. And like I said, I only really measured it in the post-surgery individuals on the far right. So I had to compare against pre-existing data sets in the literature. So you can see those from Cox et al. 2018 and Cox and Gillies 2020. So those are normal toads. Their nervous system is totally intact. And what we see is that our average value is actually shifted down quite a bit. It's actually half the amount of time. So our toads after the surgery where we cut that sciatic nerve and they lose the information from their legs, they're actually turning on their forearm muscle half as long, right? And so that muscle, right, is what helps to extend the elbow. So if it's on for half the time, it actually could explain why we see the rate of the elbow extension is so much slower, because the muscle that's driving that extension is only on for half the normal amount of time. So the main conclusion of this study is that toads actually are using sensory information from their legs to then inform their arms for how to land. So this is actually a really novel finding that I thought was really cool to see. And then also, right, because we caused these toads some injury that they actually never fully recover that sensory information from, this shows that the nervous system is capable of reorganizing, right, figuring out new solutions to still have those toads land effectively. So this kind of shows even if we end up having some type of injury that causes neural damage, right, that could be long lasting, there actually are ways we potentially could recover our overall behavior or movement. So that kind of concludes actually the last chapter of my dissertation, but I actually didn't stop my research there. I'm really interested in how humans could potentially use their upper body to help them move effectively. So I specifically looked at this shoulder rotation or thoracic rotation that people have, and I wanted to see if that affects our movement. So I'm going to bring this back a little bit to my track experience. So I remember when I was running around the curves in high school, this track, my coach would always yell at me, you know, relax your shoulders, stop swinging your shoulders so much, you know. And as soon as I got onto the straightaways, he'd be like, nice job, shoulders look super relaxed. And all that had happened, I hadn't changed anything. All that had happened was the orientation my coach was looking at me, right? When he was seeing me from the side and I was swinging my shoulders, he saw a lot of rotation. Versus seeing me from straight on, that rotation isn't as apparent. So I kind of got interested in whether this shoulder rotation or thoracic rotation actually is important for our movement. So there's this idea called the kinetic chain theory, which some of you may have heard about, that essentially core muscles can actually transfer energy and momentum out to more distal segments. So in this case, there's a slow motion video, right, of a pitcher playing where he's going to wind up his trunk or his chest, right? And eventually he's going to transfer that energy and momentum in his chest into his arm, down his arm, out through his hand and into that ball, right? And so the same principle can be applied to kicking objects or even putting a force into the ground, right? Right. So in addition to that, because our shoulders and our hips, right, our torso are rotating, and they have mass, they're going to act like torsional pendulums. So they have angular momentum to that. And so normally we walk opposite arm, opposite leg, and those angular momentums cancel out, and we can walk in a really nice straight line. But you can also walk same arm, same leg, but those angular momentums do not cancel, and it's really hard to walk in a straight line because you get this torque about the center of mass. And the last thing is that it just looks and feels good, right? So there's probably a reason why we rotate or strut a little bit, right? And everybody's not going to strut like these actors in Zoolander, right? So clearly there's this Goldilocks zone. There's some amount of rotation that's probably the best, right? And you don't obviously want to stay super rigid, but you don't want to be rotating around or strutting around when you're just walking down the street. So what I did, and I did a couple different conditions, but in each condition, I basically had 10 subjects split equally between the sexes. So I'm really interested in the metabolics, right? The energetic cost of this movement. So we measured the VO2, which is the volume of oxygen that they're consuming per unit time and per body mass. And that really gives you a metric of how much energy it takes to move. And similarly, the CO2 is the amount of CO2 you're producing, and that should be roughly equivalent to the VO2 if it's aerobic exercise. And then with this mass system, you can see me wearing on the left, you can actually also measure heart rate as well. So I actually tracked this data in the matrix, which was really cool because I got to meet Keanu Reeves. Just kidding. Unfortunately, I didn't get to meet him. This is with Vicon. It's a motion capture system that basically allows you to track body position, impact forces from that treadmill that you can kind of see popping up there, and muscle activity as well if you want. So here's me kind of instrumented up on the treadmill, and you can actually see I'm holding my sides with my hands. So the reason we did that is because if you ask people not to rotate their shoulders at all, you can still actually generate angular momentum just by swinging your arms, right? So we wanted to control for that. So I had subjects kind of hold their side while they're doing these different conditions. And we had 3 kind of major conditions that we tested. There's the preferred rotation. So you're looking down on a subject and you can see the markers on their shoulder there, and they're going to sway a little bit when they prefer to walk. So this is just their normal walk. We ask them not to rotate, so try to hold their shoulders as rigid as possible. And then we ask them to really force that rotation, right, and strut their stuff. So when we look at walking here, we're just looking at the amount of rotation we see across these different conditions on the bottom. And so this is a slightly different graph because I only have 10 individuals or 10 data points, so it's really hard to draw that bell-shaped curve, that cloud that we saw before. But this is a box and whisker plot. It's really similar. So this red central line, that represents the median value or the middle value. And in normally distributed data, that should be pretty close to the mean value or the average. The blue boxes represent the middle 50% of the distribution, and these black lines or whiskers kind of represent the min and the max values, unless, of course, we see outliers, which we do in the two preferred conditions. We see these two little red stars. So clearly someone really likes to rotate when they're walking, and I'll let you take a guess at who that was. But so when we look at these different conditions, I'm actually going to focus on the third condition first, which is their preferred pre any trials or asking them to not rotate or force the rotation. So we actually had them walk at their normal amount first. So that's that third condition there. And then we randomized whether subjects were asked to not rotate that first condition or really force that rotation, right? And so because we exposed these two, we asked people to try these two kind of weird conditions, we then again asked them to see what their preferred amount of rotation was after they experienced both of those. And so we can see in both the preferred conditions, it's actually much closer to the no rotation. It's really hard to get zero rotation when you're walking. But so it's really close to that no rotation. And there is a little bit of a jump right after they've experienced those two. But unfortunately, because I only have that small sample size of 10 people, I can't really say anything statistically about that small jump there. And obviously the forced rotation, right? People can rotate quite a bit. So I'm really interested in figuring out behaviors that this rotation might be more important, where we get a larger jump in the preferred amount of rotation. So what I actually did was I asked subjects to go from a walk to run transition. And now they can freely move their arms, but I wanted to see how much they naturally rotate their shoulders. So here on the x-axis, you have speed. On the y-axis, you have the amount of rotation. And I've actually broken up each subject's walk and run. So the walk are all the little circles. And when they start to run, they're going to turn into squares. And here, the different colors represent different individuals. And no, red and blue doesn't really represent the difference in sexes. They're just different individuals. But what we can see here from these different lines that are plotted is that those slopes actually significantly differ from zero. So what this means is that people are actually increasing the amount of rotation as they increase in speed for both walking and running. So again, like I said, I was also interested in these different conditions. So I wanted people to walk uphill, right? Because that requires more energy. So again, here we're just looking at the amount of rotation. So again, no rotation is really low. The forced rotation is pretty high. And we measured the preferred amount of rotation after those two conditions. And we can see that it's a little bit higher than on that last condition when we dropped the treadmill back down to level. So clearly people do like to rotate a little bit more walking up slopes. And then I also checked running, right? Because that's originally what I was interested in. So again, we just checked their no and forced rotation and then what they preferred to rotate when they run. And we see that it's much higher, around 25 degrees when people are running. So this is the same information you just saw. Now, instead of having just a single red line, we're having a red bar that represents the median value or the middle value, right? And that blue error bar represents the middle 50% of that data. And this is just for your reference to see how people are rotating, because we don't really care how much they're moving. We care about how much energy it takes to move, right? So here in this graph, you're looking at the VO2, the amount of oxygen they're consuming, and essentially the amount of energy that they're using. So this dot or dashed red line at the bottom represents the resting metabolic rate. So when you're walking, you're actually increasing your metabolic rate about 3 times. And we can see they're all pretty low. The no rotation is actually lined up with those preferred rotations. And that makes sense because we don't really rotate all that much while we're walking. There is a little bit of a bump in the amount of energy that it takes to really force that rotation or strut while you're walking. And then what's interesting when we look at the incline data, we see the no rotation, the forced rotation, and the preferred amount of rotation on that incline is actually almost identical. Those distributions and that median value looks the same. And then of course that last value, when we drop the treadmill back down to a level condition, it cuts the amount of energy it requires half. And then finally, in running, we see kind of a weird trend again. Actually, that forced rotation has kind of shifted downward. And we think the preferred rotation is also going to be shifted downward. We just have two kind of outliers that really dropped the amount of energy demand they needed to run. So clearly, this rotation may actually help or provide a benefit, at least at higher speeds. So I kind of want to summarize what I've shown you today. My first chapter, I really showed that these toads are landing the same way, right, across these different surface conditions, whether it's rigid like concrete or compliant like a springy trampoline. And then in my second chapter, I actually showed that they're coordinating or tuning their muscles the same way across those different compliance treatments. And then in my third chapter, right, I showed that it was actually the sensory information from those legs or the hind limbs that actually was informing how the arms are preparing for that landing behavior. And then in that last little passion project of mine, I essentially showed that this rotation might actually, that shoulder rotation might actually have a role or play a role in our movement or walking more efficiently. So with that, I want to thank all my funding sources, all my committee members, specifically Manny and Zizi, my doctoral thesis advisor. Yeah, he really gave me a lot of intellectual freedom to pursue the projects that I was interested in, and for that, I'm super grateful. I want to thank Matt McHenry and Monica Daly, my core committee members, because they actually opened up their labs and provided resources and equipment to actually allow me to collect some of the data that I presented on today. I also want to thank Jim Hicks and Vince Caiozo because they were on my advancement committee and really helped shape the direction of my dissertation early on. Of course, I need to thank the undergraduate researchers that helped me. So some of them are actually here today or tuning in. This was a fantastic team of people to work with. They were amazing people. Actually, the first three ended up developing independent projects of their own with me and turning those into their honors thesis, which is really cool to see. So I'm grateful for them because honestly, I wouldn't be able to present the work I've shown today without them. And then I want to thank everyone in the ZZ Lab, both past and present members, because really over the past 5 years, they've helped me develop as a scientist tremendously. So thank you to all of them. I want to thank Zan Cox specifically because her research was really what paved the way for my work. We overlapped a little bit here while she was at UCI, and she was like a tremendous mentor for when I was kind of going through that third chapter and even just with all my work. I also want to thank Amanda Barth, who actually went into the field with me to collect some of these specimens that I showed you the data for today. And then, of course, I need to thank Sasha Voloshina and Daisy Vega because they really helped me navigate the matrix, right, and learn how to analyze that human motion data in Vicon. And I need to thank the Daily Lab. Unfortunately, I don't have a picture of you all, but we kind of have combined lab meetings, and they've provided a wealth of feedback on my presentations and my research over the past several years. And then, of course, the UCI Physiology Group or 210 group, so a lot of you are all here today. Again, they provided a lot of professional development and feedback on talks like these throughout my time here. I also really want to thank Bob Narsession and Janet Wheat, my two mentors here on the left, who actually got me interested in biomechanics at a really young age, like when I was in high school and throughout college. And I honestly probably would not be in this program today if it weren't for them. So I definitely want to thank them. And I want to thank all my friends for basically helping me get out and explore Southern California, getting away from school for a little bit and just going on some fun adventures. I also want to thank my family for their continued support and unconditional love. They've come out to visit me a bunch of times while I've been out here, and yeah, they've always supported me since day one. I also want to thank my wonderful partner, Elizabeth, who baked all the cookies for everybody that came in person. So please grab some. I see there's some left. And she really helped me survive, especially during the pandemic, and even came in, you can see on this picture over here, to collect research with me during the middle of the pandemic, the peak of the pandemic, actually, when it was like me and one other person allowed in the entire building. So a lot of this work couldn't have been done without her, and I'm grateful for her as well. So with that, I want to thank everybody else who's here and attended in person and on Zoom. I see you too. And with that, I'll take any questions.